and inbreeding depression. So when or if the time comes that we can reintroduce species to the wild, we then have genetically diverse offspring and not inbred. All right. So non-random mating is something that is a very big deal in small populations. We're going to talk about drift a lot. Genetic drift is the phenomenon of allele frequencies changing due to chance events in a small breeding population, usually resulting in a loss of alleles. So if chance events, so here is genetic drift. So let's say this is the last surviving group of green and beige beetles. If we had 1 million of them, and the green ones are males, if we had 1 million of them, losing 2 males would not be a big deal. But if we have 9 left and we lose 2 males, that means there's 1 breeding male. That's a really big deal for a small population. So chance events that eliminate genetic diversity are a really big deal in small populations. And this is a huge issue with endangered species. So you guys are going to do a project on poaching. I'm going to get it together in the next couple of days, and we're going to look at these issues and how we deal with this as an international problem. Right? So we just lost white rhinos in 2018. There's no more of them. They're extinct, which is truly tragic. But we're going to lose a lot of our other charismatic animals, like southern white rhinos. Right? So the northern white rhino is now extinct. We're going to lose black rhinos. We're going to lose elephants. We're going to lose hippos. If we can't get this poaching thing under control. And even if we save these animals from the brink of extinction, they're going to have really, really small gene pools. So what do we do with that then? That's a big issue. So, so these are all important things to think about. So with drift, there's 2 big issues that cause genetic drift. And these effects are called founder effects and bottlenecks. So you can have population. So this is the founder effect. So the founder effect is when, so this is slide 29. The founder effect is when you have, so this is the ancestral population. And then you have, what happens is you get a small sample of an ancestral population. And that ancestral population is not, the genetic diversity is not representative. Right, so this small subset of the ancestral population, the diversity is not representative of the old world's population or the ancestral population's genetic diversity. And then so these founders migrate and then they colonize a new land. So the genetic diversity of the population that grows from that area is representative of the migrants, not of the original population as a whole. So any sort of genetic abnormalities get skewed in this new population as a result. So a good example of this is in old world Jewish individuals and new world Jewish individuals, and a subpopulation of Jews called Ashkenazi Jews. So in the old world, meaning Eurasia, okay, Ashkenazi Jews have low rates of Tay-Sachs disease, so you can use this as Tay-Sachs. What happened was that there was a founder population of individuals from the old world that colonized the new world, North America. And there were a few individuals that had Tay-Sachs. So as a result, new world Jewish Ashkenazi Jews, they have a higher prevalence of Tay-Sachs. So that's due to the founder effect, where what happens is you squeeze the population and then you regrow the population, right? And this is due to migration. So bottleneck does, right, so if we look at endangered species, right, elephants and rhinos, right, there's 30,000 rhinos left. Elephants, there's 450,000 of them. There were, at the start of the 21st century, right, there were over 10 million elephants in Africa, and now there's 450,000 of them. So even if we save that population, we have created a bottleneck. So what happens is, these are cheetahs, right? Cheetahs have actually come back from the brink of extinction. It's been quite successful. So we've managed to deal with the poaching with that. And if you look at things like rhinos and elephants, they have ivory. So ivory is a big international business now. So they are in demand. fur isn't so much in demand anymore. So you can see what happened, right, is what happens in a bottleneck is you have an ancestral population with a great amount of genetic diversity, and so these different colors represent. And then you force that population to the brink of extinction. So you squeeze the population numbers, and then the population survives. But the population no longer has the diversity that it originally had. So this is why having habitat that is suitable for organisms is important for preserving organisms. Because even if we bring back the numbers, they are still really vulnerable because they don't have the diversity to deal with changing environments. So think about poaching / climate change. So environments are changing really quickly. We know that if environments change quickly, then we have extinction. Top that off with the lack of genetic diversity. We're at a very big turning point in world history when we talk about extinction right now as a result. So this is why we need zoos. So zoos house an important stock of genetic diversity for critically endangered animals. And this is a great example. This is something that I worked on when I was at the Calgary Zoo. So these are red pandas. Red pandas were almost extinct. And the Calgary Zoo was one of the world leaders in captive breeding programs. And now we have a sustainable population in China, again, of red pandas, where they're native from. They were extirpated from that region. So they may not be everyone's favorite cup of tea, but unfortunately they're necessary with current, with the current human population being as large as it is and exhausting Earth's resources to the level it is, right? So genetic drift, right? So this was a bottleneck, and we've regrown, and we've tried to maintain the diversity as much as possible. Okay, so last big piece here, right? So natural selection works on diversity, right? So when the environment changes, it's good to have diversity because there'll be different characteristics that will be advantageous for survival. This is, again, why we want to have diversity, because environments are constantly changing. If you don't have diversity, right, what is advantageous tomorrow might not be advantageous today. And if you don't have the diversity to handle that, then populations can't adapt, right? So coupled with natural selection is sexual selection, right? So not only do we have a drive to survive, but we have a drive to reproduce, right? So there's this trade-off between natural and sexual selection, right? So if I were to look at a continuum, right, we have natural selection and then we have sexual selection here. Right, so there's this tug-of-war between the two of them when we talk about adaptive traits, because sexual selection, right, leads to things like ornamentation in males, where they have different types of organs and physical representations or elaborate mating displays that are actually a fitness cost. They're very expensive to maintain, and they make individuals vulnerable to predation. But if they can maintain those characteristics and still survive and thrive, then they must have a genetic advantage. Natural selection is that drive to survive, right? So if I'm a lynx, for example, I constantly have microevolutions occurring to make me more buoyant over the snow in order to catch snowshoe hares or camouflage better. And in response, right, the snowshoe hare is constantly having microevolutions to be able to outrun the lynx. So that survivability, but let's think about this, right? If the lynx has a large piece of ornamentation, like large ear tufts, for example, that male lynx have, that makes them more noticeable. So it might mean that they're not able to collect food as easily as a smaller female. But if they're able to collect food and still be able to maintain that expensive characteristic, then there must be some sort of genetic advantage. So there's this tug-of-war between the two of them. So you can look at my slides, right, with sexual selection, fitness, right, and again, female, right, if we look at female goals, and we talk about evolutionary fitness, quality is the big deal. Males, it's quantity, right? So males generally, because they are chosen by the female, they're the more sexually selected sex, and that's why there's things like rutting seasons and big types of ornamentation on males, right? Here's the sort of one of the ultimate features, like the elephant seal, right? And sexual dimorphism too, right? Males in heavily sexually selected species are often much larger, and sometimes it's the opposite. In hyenas, for example, females are twice the size of males, and they compete for males. In black widow spiders, right, the female is much, much larger than the male. The male's tiny, and then the male is consumed by the female while they mate, and things like that, right? So why are tigers orange for sexual selection, right? Their prey is colorblind. So they can have that nice, healthy coat in the middle of the taiga, which is coniferous forest, right? And snow, where they stick out like a sore thumb. But if they can maintain that beautiful, nice coat in that scarce environment, then they must have superior genetics, right? So tigers are orange because of sexual selection. Right, so there is the sexual selection piece with humans. I'll leave that slide for you to read. There's a little bit of tongue-in-cheek humor in there. So natural selection, again, sometimes we select for traits that are advantageous in different environments. Sometimes there's a heterozygous advantage as well. So we select for the dominant and the recessive trait. And that's why recessive traits still persist, because in some cases they're actually advantageous. And a good example of that, so you saw cholera there, but another good example is sickle cell anemia. Okay, so in normal red blood cells, right, normal red blood cells are red when they look like that, but sometimes when they, right, this is a sickle, right? In cases where individuals have sickle cell anemia, the red blood cells look like this, so they can't carry oxygen. Now, what happens in areas that are malaria-stricken, right? Mosquitoes transmit the malaria protist. Now, these guys have reduced motility. So these sickled cells, and these are normal motility. So these are RBCs. Now, what happens is the immune system wipes out the sickled cells. In areas that are malaria-stricken, like in the tropics, it's actually advantageous to be heterozygous for sickled cells. So this gives you the sickled cell genotype, and that gives you the normal. This is called codominant because both traits are expressed. Now, that doesn't happen all the time, but it does happen in this situation. So in areas that are malaria-stricken, it's actually advantageous to have some sickled cells and some normal cells. And that's because when the mosquito infects the individual with the protist, these can't move that well, so the protist attacks these sickled cells and leaves these ones alone. So even though oxygen transmission through the body is reduced, you still have normal cells that work. Whereas here, the immune system wipes out these cells. So the cells that are getting wiped out by the immune system are the ones that are carrying the protist, so it doesn't compromise the body's ability to transport oxygen. So that's called heterozygote advantage, and so that's where some recessive traits might be selected for. Okay, so those are your main things that cause gene pool change, and these will cause Hardy-Weinberg equilibrium to not be met. And as a reminder, right, Hardy-Weinberg equilibrium is pretty much never met. But what we try to do is study populations to figure out what's driving that change the most. So I'm going to give you a couple examples in your key that look at the theory behind Hardy-Weinberg equilibrium. Okay, so Ecology starts on page 121 of your text. So I'm actually going to turn to page 127. So some good questions here that deal with Hardy-Weinberg. Here's a good one. So the Amish are a group of people who rarely marry outside of their community. In one group of Amish in Ohio, the incidence of cystic fibrosis was 19. Second group of Amish in Ohio had no affected individuals. No members of either group are closely related. These data illustrate genetic drift or the founder effect. So the founder effect seemed to occur when a non-representative subpopulation forms the basis for a new population. Right, so we have a sample group from an ancestral population. They colonize a new land, so the new colonizers reflect the genetic diversity. Right, so the new population reflects the genetic diversity of the colonizers, not the ancestral population. Now I'm going to look at these equation questions later. But what Hardy-Weinberg allows us to do is quantify gene pools. So the frequency of alleles and the frequency that these alleles exist in different genotypes. Page 128 are all questions that I'm going to address in the actual Hardy-Weinberg lecture. Okay, this is also a good one, number 5. The answer is this. This represents Tay-Sachs. Okay, so Tay-Sachs disease is a hereditary disease that kills 1 in 360,000 individuals in the general population, but much more in the Ashkenazi Jew. The disease disrupts or halts proper formation of lysosomes and inhibits fat deposition. Studies suggest that, so this is heterozygote advantage. So if you are heterozygous for Tay-Sachs, it gives you protection against TB. So TB is not really a problem in the world today, but if it became more common again, you would expect the Tay-Sachs allele to increase because it gives a selective advantage to that, right? So take a look at that. I'm not going to talk about those questions. So those are all things directly related to gene pool change. This question, page 137, Challenger question number 2, is also a really good question. So the fathead minnow is a small fish common in Alberta waters and is used as a food source by many different predators. When injured, some minnows secrete a chemical called Schreckstoff that both attracts predators and causes other minnows to huddle in large groups. Approaching predators tend to be distracted by the mass of minnows and by each other. Often the injured minnow can escape. So this gives selective advantage, all right? So that's a mutation that arises and then it's going to be selected for. So you would expect, right, the frequency of the gene that controls the production of Schreckstoff should increase in the gene pool because it gives, so the ones that are going to reproduce are the ones that survive, right? So they're not going to get picked off, so they're more likely to have that trait. Take a look at page 138, question 3 as well. And this is about herbicidal resistance, right? So as we use more pesticides and herbicides, they become resistant quicker. Challenger question 4 on 138 is very good as well. So these are, again, all related to Hardy-Weinberg equilibrium. I'm going to leave those there for you. If you have questions about them, please let me know. But anyway, that's the theoretical lesson for today. I will post another video in the next couple days, actually looking at how to use the Hardy-Weinberg equilibrium equation. But hopefully this is helpful. We will chat soon. Bye now.